Thank you for the kind introduction. I'm Hyung from the Bioinformatics Lab at the Yonsei University. First, I would like to express my sincere gratitude to MVIF for selecting my work as a highlight talk. And today, I'll be presenting my recent work titled High Quality Genomic Catalog of the Human Oral Microbiome Broadens its Phylogeny and Clinical Insights. The human oral microbiome plays a central role in maintaining oral health, and its dysbiosis is known to be directly linked to oral diseases such as periodontitis and dental caries. And more recently, there have been an increasing evidence which suggests the association of human oral microbiome and several systemic diseases such as colorectal cancer and inflammatory bowel disease. However, a comprehensive genomic catalog capturing the true diversity of the human oral microbiome has been limited, as existing databases have been largely based on the isolate genomes but include only a limited number of metagenome assembled genomes. To address this limitation, we collected existing publicly available human oral metagenomic shotgun sequencing datasets and constructed Human Reference Oral Microbiome Catalog, HROM. For the construction of HROM, we collected nearly 8,000 oral WMS datasets from diverse countries and diverse oral subsites. And we processed them using an up-to-date assembly pipe and binning pipeline. And in total, we were able to recover over 140,000 medium quality genomes, and including more than 72,000 near complete genomes. From these, we were able to obtain 3,426 near complete species level genomes, and only 14% of these species contained isolate genomes, and approximately 59% lacked any species level classification, which underscores the previously undescribed diversity of the human oral microbiome. Comparing with the existing oral catalogs, HRM substantially contains more genomes and more species. And also, using independent validation human oral WMS datasets, we confirmed that HRM has the highest metagenomic reclassification rate. Using HRM, we also identified oral Patescibacteria subclade associated with periodontitis. When we compare the oral Patescibacteria, a phylum belonging to the CPR superphylum, with publicly available bacteria from other sources, oral members formed a distinctive phylogenetic and proteomic clade clearly separated from the environmental counterparts. Also, protein presence absence-based clustering of species revealed a subclade, cluster 16, that showed a significant enrichment in periodontitis samples from saliva. Also, finally, when we used the relative abundance of the cluster 16 for the disease prediction, it performed comparably to P. gingivalis, a well-known periodontitis pathogen. Finally, through comparative analysis with the Human Gut Microbiome Catalog HRGM version 2, we identified 330 oral gut share species. And among these, we additionally defined 42 ectopic oral species, the species that are highly prevalent in healthy oral microbiome, but rarely detected in healthy gut microbiome. And across 12 public colorectal cancer gut microbiome datasets, we found that the most species enriched in gut microbiome of colorectal cancer patients belonged into the ectopic oral species or the oral gut share species. And moreover, the summed relative abundance of the 42 ectopic oral species were significantly enriched in the gut microbiome of diverse systemic diseases and provided a notable predictive power for the disease classification as well. And HRM is currently published at the November issue of Cell Host and Microbe, and resources including all assembled genomes and custom reference databases for Metaflon and Kraken2 are available in our website. And lastly, I would like to thank my all lab members for the great support, and principal investigator and Lee for the supporting and guiding my research. Thank you for listening, and I'll take any questions.